Go ahead.
Hi, my name is Megan Lanius and my partner is Charlie Geyer. We will be showing you all how to fill out Form 6A, the Potentially Hazardous Biological Agents Risk Assessment Form. This form is not required for everyone, only those who are using biologically hazardous agents such as microorganisms, rDNA, fresh or frozen tissue, blood or blood products, and body fluids. These must be approved by the SRC before any research begins. This form is also to be completed by the qualified scientist or the designated supervisor in collaboration with the student researchers. Page 18 of the ISEF Rules and Guidelines Handbook, the Potentially Hazardous Biological Agents Risk Assessment, which we will cover later, includes examples of potentially hazardous biological agents. Now, I will let Charlie discuss the next section of the form.
Thank you, Megan. Here we are at the top of Form 6A. Most of the questions on this page will refer heavily to the information on page 18 of the ISEF Rules Handbook, which we will look at on the next slide. Page 18 covers how to do a risk assessment for a project containing these biological agents. You begin this form by writing all the students' names and the title of the project just as they appear on all the other forms. Now, as Megan mentioned, this is to be completed by the qualified scientist or the designated supervisor with the students. The question blanks on the original form are small, so you are permitted to attach additional pages to adequately address each question. Section 1 is the project assessment. As mentioned, page 18 will cover how to perform a risk assessment for the project. Question 1 begins by asking the qualified scientist or the designated supervisor to identify the potentially hazardous biological agents to be used in the experiment. Include the source, quantity, and biosafety level of each microorganism. Be sure that the experiment is well planned out because this form must be approved before any experimentation begins. This is to say that you cannot add any microorganisms to the experiment after this form has been approved by the SRC. Question number 2 asks to describe the site of experimentation, including the level of biological containment. Let's take a look at page 18 to define the biosafety level risk group as asked for in question number 1, and the level of biological containment asked for in question number 2. This is page 18, the potentially hazardous biological agents risk assessment. This page has 3 components. The first walks you through the process of conducting a risk assessment. Your goal is to first assess the risk group for the biological agent being used, then decide which level of containment is available for the project, and then consider the training of the qualified scientist or designated supervisor. Then make a final judgment on what biosafety level the research needs. The second component details the classification of biological agents into risk groups. The risk group should already be determined for each biological agent, as these are commonly accepted categories for microorganisms. Biosafety level 1 risk group has the lowest risk. Some examples are listed here. Biosafety level 2 risk group has moderate risk and has more restrictions for the environment in which it is researched. Some examples are listed here. Any biological agent with biosafety level 3 or biosafety level 4 risk group are prohibited for ISEF. Starting on page 16 of the handbook, more examples and information is given to classify the biosafety level risk group of biological agents. Megan will explain the third component of this page, the levels of biological containment.
Thank you, Charlie. Here we can see the levels of biological containment that are required for different biosafety levels. Biosafety level 1 laboratories require work to be done on an open bench or in an appropriate biosafety hood. Decontamination can be achieved by treating the agents with chemical disinfectants or by steam or autoclaving. Lab coats and gloves are also required for this biosafety level. In a biosafety level 2 laboratory, access to the laboratory is restricted, meaning that individuals need a certain clearance or approval to enter the lab. Personal protective gear, such as lab coats and gloves, are required. Protective eyewear is to be used as needed. For disposing hazardous agents, an autoclave should be accessible. This biosafety level also needs to be supervised by a scientist who understands the risk associated with working with the agents involved. And like Charlie said, biosafety levels 3 and 4 are prohibited regarding ISEF rules. Now, back to Charlie.
Thank you, Megan. So after the level of biological containment has been determined for the experiment, you must be sure that lab facilities are adequate for that containment level. Use this information to fill in question number 2. Moving on to question number 3, this question asks to describe the procedures that will be used to minimize risk. This part on the form does not provide a lot of space to answer, so please don't be afraid to use extra pages to thoroughly write down procedures and materials used. In sentence form, please list all materials and procedures used to minimize risk. These will often coincide with the level of biological containment. Examples include gloves and eye goggles are required for ... Lab coats must be worn during. CAV hood is used while, etc. Question 4. What final biosafety level do you recommend for this project given the risk assessment conducted? Biosafety level is based on risk group of the biological agents, level of containment the student has available, and the experience of the qualified scientist/designated supervisor. An example answer to this question may begin like, considering that none of the microorganisms exceed risk group 1, that the students have access to a biosafety level 2 lab, and the qualified scientist has experience working with a biosafety level 2 biological agents, I recommend this project be classified as biosafety level 1. The last question in section 1 asks to describe the method of disposal of all cultured materials and other potentially hazardous biological agents. Here's a snippet of the ISEF rules and guidelines. In short, you must ensure that all disposal is done in an environmentally safe manner and in accordance with good laboratory practices. This should also be in sentence form, such as, used gloves will be deposited in a designated biohazard disposal container, which will be handled per lab protocol. Megan will now discuss section 2 of Form 6A.
Here is the training section of Form 6A. Under number 1, the qualified scientist or designated supervisor will record the necessary amount of training that the students need in order to work with their potentially hazardous biological agents. An example of what would be included under this question would be the training received for the specific laboratory and biosafety level. Each laboratory typically differs on their training requirements. Under number 2, the experience and training of the project supervisor as it relates to the project studied should be listed. For example, if a teacher majored in microbiology and has experience in a biosafety level 2 facility, that would be noted. Next, I'll let Charlie wrap us up.
Thanks, Megan. Section 3 is to be used for all cell lines, microorganisms, and tissues. Read the 3 bullets and check which applies. These relate to the facilities used for the experiment. Per the first box, if the research is not conducted at a regulated research institution but will be conducted in another lab, be sure to check what level of containment corresponds with that lab. Per the second box, if the research is at a regulated research institution that requires pre-approval, you must include the institutional approval forms. Per the third box, if the experiment is conducted at a regulated research institution but does not require pre-approval for the type of study, check here. Sign and date this portion of the form. Section 4 is for SRC approval only. Do not fill out this section. A reminder that if an experiment is using potentially hazardous biological agents, then this form must be filled out and approved by the SRC before experimentation begins. Thank you for watching this video. Happy experimenting!